So today we are going to solve some of the questions from the chapter that is molecular basis of inheritance. And probably you guys were not in the last class. So today I have chosen some of the questions which are basically appearing on a repeated basis, especially in the past 8 years. Okay. So these questions are important and all of them are previous year questions. So let's see how your knowledge is based on your understanding. Okay, so let's get started. So we have the first question here. Unequivocal proof that DNA is a genetic material was proposed by. So you have our following options, that is Wilkins and Franklin, Frederick Griffith, Alfred Hershey and Martha Chase, Avery, Mecklin and McCarthy. Okay, so these are your options. So I launched the poll for you. So there is an MCQ poll, so you can answer your questions. Let's see what you have. And don't worry if you get it wrong. Everybody, all of us make mistakes. It's fine. I'll read the question again. Unequivocal proof that DNA is genetic material was yes. First proposed one. Perfect. Nice. So your answer you've already given, okay, or 2 on 2. So let's end the poll. So you guys are right? So you have chosen your option as option C, that is Alfred Hershey and Martha Chase. So Alfred Hershey and Martha Chase, so they perform an experiment. We already know that that they were the first one to completely give an experimental evidence that DNA was a genetic material. So in their experiment, we know that the organism and the experiment model they chose was bacteriophage and E. coli. So bacteriophage, they label it by sulfur and phosphorus radioactive isotopes. And then finally, they get that pellet and supernated, right? So when the pellet, that is a cellular part, was radioactive, that showed that there was DNA material that was transferred between bacteriophage and E. coli. So that is what led to the proof that DNA is a genetic material. So you guys are absolutely right. Option 3 is the correct option. So when you look at the other side of the story, that is Avery, Mecklin, and McCarthy. So these 3 scientists, as we had discussed earlier, they were very exceptional in giving us, and almost a proof that DNA was a genetic material, right? So they worked on the Griffiths experiment and they tried to understand the biochemical nature of Griffiths experiment. And unfortunately, many of the biologists could not approve or accept it because of exceptional simplicity of DNA molecule. But once Alfred Hershey and Martha Chase proved this, so it changed the entire notion about how we approach the DNA. Anyways, nice answer. So let's go for the next one. ESTs, that is express sequence tags, refers to. So your options are certain important express genes, all genes that are expressed as RNA, all genes that are expressed as proteins, all genes whether expressed or unexpressed. So I will launch the poll. So let's go. ESTs refers to. What are express sequence tags? Don't you think that this is quite a direct question? It is. Okay, good. Any more options? Okay, we are almost like 5 seconds out. So I'll end the poll now and let's see what is the right answer. Okay, so you're right. So the option that you have chosen is B, that is all genes that are expressed as RNA, and this is absolutely right. Okay, with respect to express sequence tags. So nice, good work. So anyways, you've chosen the right option, but Now, maybe one of you here was not able to find the right answer. So here is the explanation for this. Express sequence tag. So basically, we get to know about this term when we are talking about Human Genome Project, isn't it? So we understand that in this HCG, that is, Human Genome Project, HGP. So here, there was a huge amount of gene that was supposed to be annotated, or the genes to be decoded, right? So at that point, a blind approach to study each and every single base pair was taking a very lot of time. And our scientists thought that if we target the express genes, so DNA can first express itself as an RNA, and RNA is a very important, what do you tell, intermediate situation. Okay, so these tags that were used for the genes that are expressed as RNA are called as express sequence tags. So basically, we target any gene that is expressed as RNA. So that is what express sequence tags are for. So basically, we use this express sequence tags to locate and annotate the genes that are present in our chromosomes. So this is option B, and you guys are absolutely right. So let's go for the next question. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? So we have the options: transcription of only snRNAs, transcription of rRNAs, 28S, 18S, and 5.8S, transcription of tRNA, 5st RNA, and snRNA, and transcription of precursor mRNA. So these are our options. So let me launch the poll for you guys. Yeah. So the poll should be live now. Yeah. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? Yes, perfect. 2 on 2. So we have the quick answer. Okay, just give me a second. So I don't want to just waste the time. So what about the students from the YouTube? Okay, what's your answer? Okay, you can just put your answer in the chat box. Okay, so let's get back. So your answer, that is the transcription of tRNA, 5st RNA, and then snRNA, is absolutely right. Okay, so I'll end the poll also. Okay, is absolutely right. So basically, whenever we talk about RNA polymerase 3, so we have several options, like several questions to target on RNA polymerase. For example, obviously the first question would be what type of RNAs are what type of RNAs are transcribed by a particular RNA polymerase. For example, what you have chosen, that is RNA polymerase 3. Sorry, RNA polymerase 3. It transcribes tRNA, 5st RNA, and snRNA, is absolutely right. So when it comes to RNA pol 1, okay, so our interest shifts to it transcribing, that is rRNAs, 28S, 18S, and 5.8S. And then when it comes to RNA polymerase 2, it goes for the precursor mRNA, or it is also called as hnRNA, or especially it is asked in eukaryotes, so we'll go for precursor mRNA or pre-mRNA, right? And sometimes they will ask what type of polymerase is, what type of an enzyme is RNA polymerase. So we tell that RNA polymerase is a holoenzyme, right? Holoenzyme. So this is the type or the nature of RNA polymerase. Why? Because it is made up of several subunits like alpha, beta, and gamma in the prokaryotes. Whereas when it comes to eukaryotes, it is made up of several number of subunits, like more than 10 subunits. Okay. So this is one type of question, like what type of an enzyme is RNA polymerase, that is holoenzyme. Okay. Apart from that is what type of an RNA is specifically transcribed. So that is very, very important. So anyways, you guys got the right answer. So that's good. Sorry. That's good. So let's go for the next question. Okay. Did I end the poll? I did. Okay. So given below are two statements. Okay. The statement one is, give me a second. Yeah. The statement one is, in prokaryotes, the positively charged DNA is held with some negatively charged proteins in a region called as nucleoli. And statement 2 says, in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. In the light of the above statements, choose the correct answer from the following options below. So the options are basically, so this is your A, B and C and D. So it says statement 1 is incorrect, but statement 2 is true. Statement 1 and 2 are true. Statement 1 and 2 are false. Statement 1 is correct, but statement 2 is false. All right. So first, let me launch a poll and help you with the question. 1 second. Okay. Right. Yeah. Says in prokaryotes. Okay. That was quick. Good. Very good. Yes. I have one note. I need more. What about others? What is your opinion? Yeah. I read the options for you. In prokaryotes, the positively charged DNA is held with some negatively charged proteins in a region called as nucleoid. And statement 2 says, in eukaryotes, the negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. So these are, yeah, perfect. So I have the answer. So you guys are right. So I'll end the poll. It's about time. You answered at the right time. Okay. So nice. So first, whatever you chose in the option, that is statement 1 is incorrect and statement 2 is true. Nice. Good job. Why I appreciate you for this is because you guys have a very nice observation. You found out the mistake that in prokaryotes, positively charged DNA. Whether it is in prokaryotes and eukaryotes, we already know that DNA is negatively charged, right? So in both of the cases, it is negatively charged. But the rest of the statement, that is, if it was negative, okay, if here it was given negative, so the negatively charged DNA is basically held by some positively charged proteins, not negatively charged, positively charged non-histone proteins, especially with respect to prokaryotes. And statement 2 is absolutely right. So negatively charged DNA is wrapped around positively charged histone octamers to form nucleus. So this was the topic that we discussed in packing of DNA, right? So next one. So we have a question that is we have two statements. The first statement is the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. And the statement 2 says a transcription unit in DNA is defined primarily by three regions in the DNA, that is promoter, the structural gene, and the terminate. Okay, so let me see if you have the right option. Yeah, so the same options goes. Okay, for example, we have here A, B, C, and D. Okay. So statement 1 is false, but statement 2 is true. Okay, are you sure? Yeah, we have another option. What is that? Statement 1 and statement 2 are true. Okay, so now we have a tiebreaker. Two individuals choosing two different options. Nice. Yeah. Now we have a democracy, more votes for one option. Okay, so let's see. So I will end the poll. The statement 1, what is transcription? Transcription is when the genetic information that is present in the DNA is copied into a messenger mRNA or an RNA is called as transcription, right? So this statement is absolutely the definition for transcription. So this statement is true. Now, when it comes to the structural gene, right? When we write the diagram of a structural gene, how do we present it? We say that, okay, this is the two DNA strands, and in these two DNA strands, one strand we call it as template strand, right? And other strand is called as coding strand. So in template strand, it is going from 3' to 5', and the coding strand goes from 5' to 3'. Now here, this is where your mRNA is being synthesized, right? So here in template, we have three regions. What is the three region? One is a promoter, right? And then we have structural gene, and then we have terminate, right? So the promoter region was very highly discussed in our class. For example, I will ask you a question, so you have to put it in the chat box. Let's see if you guys remember. When we talked about the promoter region in prokaryotes, what are the regions I mentioned? Do you remember? What are prokaryotes promoters called as? And give me an example for an eukaryotic promoter, students. Use the chat box for your answer. Give me an example. I mean, like one example for a prokaryotic promoter and one example for a eukaryotic promoter. Obviously, we know the either sequence names or we know the entire, I mean, like the exact promoter name itself. So try answering this. Let's see if you remember anything. Yep. I'll wait for 10 more seconds. It's very simple. You just have to write some code words. Okay. So even then, even that is helpful. So I will just move on. So basically, when I ask you a question like, okay, name a prokaryotic promoter or a eukaryotic promoter. So usually in prokaryotes, we just discussed, okay. So we called something called as 10 regions and 35 regions, okay, if you remember. Okay. Whereas in eukaryotes, okay, in eukaryotes, we talked about Tata box, right? And then cat box. You remember? Octomer region, etc. So these are called as prokaryotic or eukaryotic promoters. So which are conserved sequences. So why I was talking about is, so usually there are many cases that this particular question has arrived with the name Tata box, and many of us will be like confused. What is Tata box, right? So we know that Tata box is a promoter which is in a eukaryotic gene. Okay. So anyways, so we have discussed a lot about this. And the coming to the second statement, that is the transcription unit of the DNA is defined primarily by three regions. That is, a promoter should be there, very important. And you know the role, it helps in attachment of your RNA polymerase to the exact site and initiate the process of transcription. First thing. Second point is the promoter regions also determine the pace of the transcription. Some genes are highly what do you tell, expressive. Some genes are do not express, they're very mellow or very low in expression. So how does this promoters help? So we have something called as upstream and downstream promoters or inbuilt promoters. So what happens as the transcription is happening, apart from the regions that we see in the initial segment of your structural gene, there are also some promoters in many of the genes that come in between the transcription process. So which might help in either accelerating the transcription, or they might break or they might reduce the transcription rate. So these are called as downstream promoters, okay, or inbuilt promoter sequences. Anyways, so we talked about termination sequences and what are the different termination sequences, and what are inbuilt termination sequences, especially when it comes to the stable hairpin loop-like structure, right? So it's all related. So anyways, whatever the statement, whatever the option that you have chosen is right. That is, both statement 1 and 2 are true. Let's go for the next question. Which scientist conducted an experiment with 32P and the 35S labeled phages for demonstrating the DNA is a genetic material? It's a very easy question and a direct one. James Watson and Francis Crick, Hershey and Chase, Griffith, and Avery, McLean, and McCarthy. So these are your options. So let's go. I will launch the poll. It's a very direct question, isn't it? Which scientist conducted an experiment with 32P, that is isotope of phosphorus, P32 or S35, labeled phases for demonstrating DNA as a genetic material? I think you guys already answered it. So, yeah, perfect. So let me not waste too much time in this. Absolutely right. The first question also asked a similar, I mean, like it was the same question, but it is put in a different sense, right? So it's a very important experiment. Moreover, that's why they keep asking this question. And your option, that is Hershey and Chase, is absolutely right. So there is no need for discussing about this. And when it comes to Watson and Crick, we know that they proposed the semi-conservative model of, sorry, they proposed the right-handed helical structure of the DNA or bDNA structure. Next, Griffith was the scientist who demonstrated transformation principle, and Avery, McLean, and McCarthy are the three scientists who worked on the biochemical nature of the Griffiths experiment. Okay, so this is the fourth one is very interesting. I think you have to go through it once again. Okay, next one. So 1 second. Yeah. Given below are two statements. Statement one says RNA being unstable mutate at faster rate. Okay. And next option is RNA can directly code for the synthesis of proteins, hence can easily express the characteristics. Okay. So you have the same options like A, B, C, D. I'll write it here, A, B, C, D. So choose your right option with respect to the RNA. Yeah. RNA being unstable, mutate at a very faster rate. Okay. RNA can directly code for synthesis of proteins, hence can easily express the characteristics. Okay. Yep. Good. Do we have more options? Very nice. Okay. So can I end the poll? So you have your opinion already. So your chosen option is option C. That is both statement 1 and 2 are true. So coming to the light of the statement 1, that is RNA being unstable can mutate faster or mutate at a very faster rate. So this is a very prominent statement of why RNA is not seen as a genetic material in many of the higher organisms because of its instability. And the main reason for the instability of RNA is its structure itself, right? So the 1 second. Okay, we have given the option. Okay. So now coming to this, RNA is, hold on. Yeah. So the answers. Okay. So anyways, yeah, coming to the light of the statement, the RNA molecular structure itself is very unstable. So why it is unstable, you guys already know, because when we compare the deoxyribose and then we compare the structure of ribose, okay, let us say this is deoxyribose and this is ribose. Okay. So this is present in DNA. So this is present in RNA, correct, right? So the sugar. So when we talk about DNA, we have a OH here and we have a H here. So what is this OH? This is in the third carbon atom. This is the second carbon atom. Here, what happens here in the RNA, here also there is OH, here also there is OH. So what is here? This is second carbon atom and this is the third carbon atom of the sugar group. Now we know that when the polynucleotide chain is being constructed, this 3' OH will react with the 5' phosphate to create a very stable structure of the DNA, that is polynucleotide chain. Here, since there are two OH, our 5' phosphate will get an option between either 3 or 2. So that will shift the structure and make it quite structurally unstable. So that is the first reason why RNA is a very unstable molecule, first thing. Second one is, and because of its nature, RNA is usually known as a catalytic molecule because of its very fast half-life. Like in a very short period of time, RNA's half-life will be done and it will become degenerative. And also because of its rapidness, it can change its nucleotides, like, it can shift, isn't it? So because of its low molecular integrity, so it can change its structure, and that leads to mutations at a very fast rate. So it's a very important thing why RNA viruses mutate at a faster rate, why RNA is not a genetic material in many of the higher organisms. All that answers because of its molecular structure. So that is right. Second option, that is RNA can directly code for the synthesis of proteins, hence can easily express the characteristics. So basically, when it comes to expression of characteristics, that is true. Once we have an RNA, it can directly code for the proteins, okay? And it happens at a very faster rate. Once a messenger RNA is created, so it happens at a very faster rate when you look at in the prokaryotes and even in the lower organisms, okay? But in eukaryotes, it takes time, but eventually it leads to the proteins once we have an RNA. So that is why both statements are correct. Both statement 1 and 2 are correct, and your option is also right, okay? So let's go for the next question. The salient features of genetic codes are: the code is palindromic, UGA act as an initiator codon, the code is unambiguous and specific, the code is nearly universal. Choose the appropriate answer from the given options given below, okay? So we have the options, that is A and B only, B and C only, A and B only, C and D only, okay? So let me launch the poll for you guys. It's an easy question. Okay, salient features of a genetic code. Okay, so you have your answer. Yes, good. We have more opinion. Come on, it's easy a question. Okay, yeah, we have an answer. So what is that? Perfect. Yeah. So anyways, so let's see what our right answer is. I'll end the poll. So we have two different opinions. One is saying option A, one is saying option D. Okay, and the majority are saying option D. So I will, let's see what is the right option. So coming to the salient features of genetic code, it is very, very obvious and it's an easy fact to remember. Okay, so when we talk about genetic codes have, following characteristics. That is, we know that there are 64 codes, okay? And in 64 codes, only 61 code for amino acids and 3 are called as nonsense, right? So that is first thing. And when you see these 64 codes, in the 64 code, 61 code for 20 amino acids, okay, 20 amino acids in total. So when they code for only 20 amino acids and we have 61 codes, we know that our code is degenerate. So what is a degenerate code? So if we observe the genetic codes, so we see that two different codes are coding for the same amino acid. So that is called as a degenerate nature of a genetic code. Next one. So when we talk about the one specific genetic code, that is AUG. AUG is a very important code. Why? Because it has dual function. One, it is coding for an amino acid called as methionine. And second one, it also acts as a start codon. We call it as a start codon. So I discussed about AUG very specifically. So usually it is not asked in your NEET level, but I also told you why specifically AUG acts as a start codon, right? So in prokaryotes, we understood that AUG codes for a special type of methionine that is called as formilated methionine, right? So the tRNA brings is also called as METF tRNA. So basically this formilated methionine, I mean, like a formalin group is added to the amino acid for a specific reason. Especially when AUG is a starting codon, that is the first three code, I mean like the first code of the entire genetic message that is present in your mRNA. So the type of amino acid that is added is called as formulated methionine. The reason is simple, because it helps in creating the fresh peptide bonds with the next coming amino acids. This is called as Fox linkage. Okay. So that is why AUG acts as an initiator molecule or initiator codon with respect to prokaryotes, or it is also called as an initiator codon. So basically in your textbook, it's not mentioned whether it is prokaryotes or eukaryotes. And now you know the reason, how it acts as a initiator codon. So it is not UGA. Okay, it's given somewhat in a reverse orientation. And the next one is, next one coming to the code is palindromic. So this doesn't make any sense, because we study palindromic sequences when it comes to biotechnology, especially the action of restriction enzymes. Palindromic sequences are the one when it is written 5' to 3' also it's the same meaning, and 5' to 3' to 5' is also the same meaning. That is like, for example, Malayalam. So that a pattern is not observed in our genetic code. So our codes are quite random actually. Okay. So what, instead of palindromic, what do we see? Our code is without punctuations. Like when we read the code from 5' to 3' direction, we do not add full stops or comma or exclamatory marks or question marks. No punctuations are there. We read it continuously. And the code is universal. And this is a very important point. Whether you take the bacteria or a human being or a protista or a human being, fungi, to any organism you just see in our living system, if AUG codes for methionine in bacteria, AUG codes for methionine even in human beings. So that is called as universality of our genetic code, which basically says that we are all coming from the same source or the same ancestry. Okay. And the code is unambiguous and specific. When we talk about unambiguous, when we have a particular statement, it do not have two different meanings. It has only one meaning. Similarly, if I say AUG is called as methionine here, irrespective of where I use this particular code, it do not code for any other thing. Okay. So that is called as unambiguous. It has a specific meaning. So these are the properties of genetic code, and your answer, that is the last two options, that is the code is unambiguous and specific, and the code is nearly universal, are the right options. So basically C and D are only. So option 4. Okay. So we have the next question. Read the following statements and choose the set of correct statements. Okay. So euchromatin is loosely packed chromatin. Heterochromatin is transcriptionally active. Histone octamer is wrapped by negatively charged DNA in nucleosome. Histone are rich in lysine and arginine. A typical nucleosome contains around 400 base pairs of DNA helix. So choose the correct answer from the given option below. Okay, so let me launch the poll for you. Yeah, sorry. Excuse me. Now the poll should be live for you guys, right? Yes, good. Very nice. Yes. So what's your option? Yes. Yeah. Perfect, guys. Okay. So can we end the poll? So anyways, you guys are right. Okay. So I'll end the poll. So your option, that is C. Okay. You have chosen, all of you have chosen option C. So option C is absolutely right. Okay. So why it is right? You already know. The first statement says euchromatin is loosely packed chromatin. Yes, euchromatin. So how do we observe euchromatin? When we do karyotyping, that is when we take an X-ray of our chromosomes, we see that there are light bands and there are thick bands. So light bands are it depicts that there is DNA is being very loosely packed, and this region is highly transcriptionally active. Okay, and heterochromatin on the contrary is a very tightly packed region and they're transcriptionally inactive. So this statement becomes wrong. Histone octamer is wrapped by negatively charged DNA nucleosome. This is also absolutely right. So histones are positively charged proteins which wrap a negatively charged DNA. Now, histones are rich in lysine and arginine. Yes, lysine and arginine are very positively charged amino acid. So the rich volume in the histone octamer or histone monomers. So that is what makes them having more net positive charge. Okay, so even the option B is right. So coming to the last option, that is typical nucleosome contains around 400 base pairs of DNA helix. We know that it is not 400 base pairs, so that is wrong. So it is 200 base pairs of DNA helix. So we have two options, that is you already so A, C, and D are only. So it's right. Okay. So let's go for the next question. DNA polymorphism forms the basis of. So we have translation, genetic mapping, DNA fingerprinting, both genetic mapping and DNA fingerprinting. Okay. So here is your options. Excuse me. DNA polymorphism forms the basis of. Yes. Very good. Very good. Yes, perfect. So you have chosen the right option. Okay, let me end the poll because we have the answer. So, excuse me. I'm sorry. Yeah. The question is DNA polymorphism. So, I think you know what is polymorphism by definition. Polymorphic DNA is highly mutative sequence of DNA, and they keep changing their order and sequence and the number of repetitions from one generation to other generation. So basically they won't follow this proper hereditary, right? Like the exact gene makeup will not be coming from one generation to next generation. So this always changing form of DNA is called as polymorphic DNA. And your option, what you have chosen, that it is used in both genetic mapping and DNA fingerprinting is right. So when we talk about genetic mapping, so in genetic mapping, we come across SNPs, that is single nucleotide polymorphic DNA, right? So these single nucleotide polymorphic DNA or single nucleotide polymorphs, so are basically used for the mapping of a specific genetic disorder or a specific gene that is quite different from the normal gene. So that is how we map an malfunctioning gene or a mutated gene. Next one, that is also a kind of a mapping. Next one, coming to DNA fingerprinting, we use VNTRs, right? Variable number of tandem repeats and short tandem repeats, STRs and VNTRs in the mini satellite or microsatellite regions. So basically, they are also helping. Okay, DNA polymorphism is the basis that helps in conducting the entire DNA fingerprinting. So without this polymorphs, it would be impossible to have a DNA fingerprint, and it would be impossible to differentiate between one individual with other individual, right? So your option, that is option D, is correct. So let's go for the next question. Which of the following is not the criteria of a genetic material? Should not provide the scope for the changes of evolution, should be able to express itself in the form of Mendelian character, should be able to generate its replica, should be stable chemically and structurally. Okay, so let me read out the code for you. So, okay, the poll is live. Here you go, guys. Okay, answer it quick. Very nice. So we have the right option. Okay, so I will end the poll. So let's see. Yeah. Yes. Thank you. So I think you're answering on the both the platforms. Well and good. Okay, very active. So, anyways, so let's get back. So I think your option, sorry, is A, right? That's what you guys have chosen. So which one of the following is not the criteria for genetic material? That is, should not provide the scope for the changes of evolution. Yes, this is absolutely wrong. So if there is no scope for evolution, it would be very impossible for the life to continue for such a long period of time. The evolution through mutation and recombination is what resulted in such diversity and such capabilities of organism to survive in extreme to diverse environments, right? So this statement is absolutely wrong. So the rest of the statement is that they should be able to express itself in the form of Mendelian characteristics, should be able to generate its replica, and should be chemically and structurally stable. Yeah, of course, all these three All these three reasons are why DNA becomes such a crucial molecule to act as a genetic material. Next one. Which one of the following experiments of Frederick Griffith resulted in the discovery of bacterial transformation? So your option is S strain, which is heat killed, is injected into a mice and the mice lived. S strain, it was heat killed, and plus the live strain, next injected into the mice, mice died. S strain injected into the mice, mice died. R strain injected into the mice, mice died. So which one demonstrates the Griffith's transformation principle? That's the gist of the question. So I will read out the poll. It's a very easy question. Yep. So we have an option. I have an answer, sorry. I'm sorry, students. Okay. So there was internet problem. Okay. Thank you. Okay, just give me a second. Yeah. Okay. So your option was two, isn't it? So your option that is S strain that is heat kill plus arsenic that is injected into the mice and the mice die. So this is the one that demonstrated the transformation principle. So this is absolutely right. So the next question. With respect to the nucleosome, which of the following statements is incorrect? Nucleosome contains 120 base pairs of DNA helix. Nucleosomes are seen as beads on the string under the electron microscope. So DNA is wrapped around the positive charge histone octamer to form nucleosome. A nucleosome is a repeating unit of chromatin. So let me launch the poll. Yeah. Sorry. Yes, correct. So now you have given the answer. So tell me how many base pairs are wrapped around the histone octamer. What is your answer? Please type in the message chat box. Do you have an answer? Yes. Yes, correct. Yeah, I agree with your answer. So I'm asking you, nucleosome contains 120 base pairs of DNA helix, isn't it? So this you're telling it's wrong. So what's the right option? I mean, like, what's the right answer? How many base pairs are bound in a DNA helix? Sorry, in a histone octamer, how many base pairs is bound? What is your answer? You didn't understand the question? So I am telling you, you told that the option one is wrong. So yes, this is the very correct answer. I'm asking you. So this one should be wrong according to you, according to me. Okay, even the textbook says wrong. So what's the right number here? So that's my question. What's the right number? You have an answer? Yes, no? So it's actually around 200 base pairs of DNA is bound, isn't it? So let's get back 1 second. Okay. So that is the answer. So your option that you have chosen, that is option one, is correct. Okay. Sagar, good job. Anyways, I appreciate your patience. In DNA, if it is, this is an interesting question. Listen, okay. If DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur, what would have been the outcome of Hershey and Chase experiment? And the options are, excuse me, and the options are no radioactive sulfur in bacterial cell, both radioactive sulfur and phosphorus in bacterial cell, radioactive sulfur in bacterial cell, radioactive sulfur in bacterial, radioactive phosphorus in bacterial cell. Okay. So these are your options. So let me relaunch the poll. It's quite interesting. If DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur, what would be the outcome of Hershey and Chase experiment? You have an option? Okay. So yeah, good. So I will end the poll now. So let's discuss this solution. Okay. So now what they're telling is, if, yeah, you have an option already. B. So both radioactive sulfur and both radioactive sulfur and phosphorus in bacterial cell would be visible. Okay, let's say if DNA contains sulfur instead of phosphorus, okay, and then proteins contain phosphorus instead of sulfur, what would have happened? Okay, now here when we perform the Hershey and Chase experiment, we get two test tubes. Okay, eventually we get two different types of test tube. Okay, so test tube 1 and test tube 2. So let's say that in the original Hershey and Chase experiment, when radioactive sulfur, that is S35, was used, now supernatant, okay, that is the watery portion, the supernatant was radioactive, correct, right? Radioactive. Whereas the pellet region that was at the bottom was non-radioactive. I'll rate it as NR. Whereas when we use P32, the supernatant was non-radioactive, whereas the pellet was radioactive. Okay, pellet was radioactive. So what is the, what does it signify? The pellet contains bacterial cells, okay? So this bacterial cell will have a cell wall, and it has nucleus, and it has DNA. And you know that the bacteriophage, they inject their genetic material into the bacterial cell, and then they get incorporated into the DNA of the E. coli. Eventually, it should be in the cell of the E. coli only. But when we perform the experiment, the pellet was radioactive, right? So if we exchange its molecular nature, that is DNA contains sulfur and phosphorus is present in proteins, what would happen? Now, here, instead of radioactive phosphorus, here we would get radioactive sulfur would be seen in bacterial cells, right? So that is the right option, option 3. Okay. So let's go for the next one. So how much time do we have? We can solve one more question. This is the last question actually. Against the codon, that is 5 prime UAC 3 prime, what would be the sequence of anticodon on a tRNA? Okay, so your options are here. 5 prime AUG, 5 prime ATG, 5 prime GTA, 5 prime GUA. So these are your options. So it's a very interesting questions. Let's see if you get it right. If a codon is UAC, okay, in the right direction, that is 5 prime to 3 prime, what would be the anticodon in the tRNA? Do you have an answer? Last question. Let's make it fast. Mahi, did you say that it is B? Okay, no, that was for prior question, isn't it? Yeah, time up. See, 60 seconds up. So can I solve this? You have an answer? Let me check once again. No, no problem. So I will end the poll, okay? So let's see what's the right option. So I don't know, like probably could have gone for one of the options. So anyways, so the answer is, the question is 5 prime to 3 prime, isn't it? So if you go from 5 prime this side and 3 prime this side, this will come G, this will come U, and this will come A. So GUA 5 prime to 3 prime, that is GUA 5 prime to 3 prime. So the option is 4. So that is how we solve it. And this is the anticodon for the tRNAs UAC. Okay. So thank you very much for attending the class, and I appreciate your patience to be there. Even there was a technical issue. So thank you very much. Okay. I'll see you in the next class, and all the best for your examinations. Bye bye. Take care. Have a nice weekend.